Good morning. So the topic for today is pulmonary surfactant. Now alveoli is the functional unit of the lung. Inside the alveoli we have a fluid lining. Also inside the alveoli we have air. So this creates a air fluid interface within the alveoli, and this can also lead to a generation of a force or a surface tension inside the alveoli. So that will lead on to the generation of surface tension inside the alveoli. Now while we are breathing, there is movement of air into and out of the lungs. Now as the air is moving into and out of the lungs, the radius of the alveoli can also change. So we need to know how this is affecting the alveoli. Okay, so that will bring on to the topic for discussion today, which is pulmonary surfactant. Now the specific learning objectives include: we will see the structure and composition of the surfactant, the source, synthesis and secretion of surfactant, functions of surfactant and its applied aspect. Now coming to surface tension. As I have already mentioned, it is a force that is caused due to the intermolecular attraction between the molecules on the surface. Now in this diagram you can see this is a diagram resembling a water droplet. So inside the water droplets you can see various water molecules. So there are surface molecules as well as inner molecules. So if you compare the surface molecules with the inner molecules, you can see that the surface molecules are always pulled inward, whereas the inner molecules are pulled in all directions. So this same principle applies for the alveoli also. Now inside the alveoli there is a specific agent that is helping to keep the surface tension at a reduced level. This agent is called as the surfactant. So surfactant is nothing but it is a surface tension lowering agent which is present inside the alveoli between the alveolar fluid and the air. Now we will see the structure of the surfactant. So primarily surfactant is a lipoprotein that is rich in phospholipids, and the principal phospholipid that you can find in the surfactant is dipalmitoyl phosphatidylcholine, which is otherwise known as dipalmitoyl lecithin. Now phospholipids, we all know that they have a hydrophilic head and a hydrophobic tail. So what is the relationship between the phospholipids and the alveolar surface tension? They are inversely related. That is, the alveolar surface tension is inversely proportional to the concentration of the phospholipids present in the surfactant. So more the amount of phospholipids in the surfactant, lesser will be the surface tension that is generated. Okay. So this is just a diagram that is showing you the structure of the phospholipids with a hydrophilic head and a hydrophobic tail. Now coming to the composition of the surfactant. As I have already mentioned, the major composition of the surfactant includes lipids. So 90% of the surfactant is composed of lipids, which mainly includes dipalmitoyl phosphatidylcholine, which accounts for 62% of the total lipids. 5% is by phosphatidylglycerol, 10% by other phospholipids and 13% by neutral lipids. Apart from lipids, you can also find proteins, which accounts for 8% of the total composition of the surfactant. And the major proteins that you can find are albumin, immunoglobulin A and apoproteins like SPA, B, C and D. And you can also find 2% of carbohydrates. So this forms the composition of the surfactant. 90% is by lipids, 8% is by proteins and 2% is by carbohydrates. Okay. So that completes the composition of the surfactant. Now from where are these surfactants produced? So in the introductory class I have mentioned about the alveolar cells. Now surfactants, they are mainly produced from the type 2 alveolar epithelial cells. They are also known as type 2 pneumocytes. So the major source of surfactant is from the type 2 alveolar epithelial cells, which are also called as type 2 pneumocytes. Now we will see how the synthesis is happening. Synthesis of surfactant is happening. So this is just a diagram that is summarizing the synthesis and secretion. So as you can see in this diagram, this is a diagram that is depicting the type 2 alveolar epithelial cells. So this cell will pick up the substrate molecule that is required for the synthesis of surfactant, and then inside the type 2 alveolar cells, it will be stored within the mitochondria and the endoplasmic reticulum. Okay. And once synthesized, they will be stored in specific bodies called as lamellar bodies. And as and when required, the lamellar bodies will discharge their contents into the exterior by a process called exocytosis, wherein they will form a meshwork-like structure called tubular myelin. Okay. And it is this tubular myelin that is forming a thin film along the alveolar surface which forms the surfactant. So we have seen you have type 2 epithelial cells, and inside the type 2 epithelial cells, the surfactant is synthesized and stored in lamellar bodies. Later on, they are converted to tubular myelin, which will ultimately form the surfactant. Okay. So coming to the steps of synthesis, the substrate molecules, which are mainly the phospholipids, they will be picked up by the type 2 epithelial cells, and they will be synthesized inside the mitochondria and endoplasmic reticulum, and they will be stored within the lamellar bodies. Okay. And then the contents can be discharged into the exterior by exocytosis, wherein they will be converted into a lattice structure or a meshwork-like structure called tubular myelin. And this forms a thin film that spreads over the entire surface of the alveoli, forming the surfactant. So that summarizes the steps involved in the synthesis and secretion of surfactant. Now we will see what are the factors which control the synthesis of surfactant. There are certain factors that can stimulate the synthesis, and there are certain factors that can inhibit the synthesis of secretion. Coming to the stimulators, so there are neural factors. For example, vagal stimulation can enhance the surfactant synthesis and secretion. Then coming to hormones, hormones like glucocorticoids and thyroid hormone will again stimulate the synthesis of surfactant. Then proteins, mainly the apoproteins, are involved in the several steps of secretion of surfactant. Then stretching of the lungs, like in case of hyperinflation, like when a person is yawning or sighing, this in turn can stimulate the synthesis of surfactant. Then drugs, for example, beta adrenergic drugs, calcium stimulants are few examples which can enhance the surfactant synthesis. And finally, exercise is a physiological condition which can stimulate the surfactant synthesis. Now coming to the inhibitors of surfactant synthesis, smoking, then hypoxia and oxygen toxicity are few factors that will inhibit or decrease the surfactant secretion. So this slide summarizes the factors that can stimulate and inhibit the surfactant synthesis and secretion. So we have seen the structure of the surfactant, its composition, from where it is synthesized and how it is synthesized, and what are the factors that are involved in the regulation of the synthesis. Now moving on to the most important part of the class, that is what are the functions of the surfactant. So in my initial slides I have told you surfactant is a surface tension reducing agent. So the first function is also it reduces the surface tension and prevents the lung collapse. Now we will see, apart from reducing the surface tension, what are the other functions of surfactant. Now another function is it helps to maintain the alveolar stability and helps in even distribution of ventilation. Now inside the lungs there are alveoli which has got different diameters. Some of the alveoli will be smaller in size, some of the alveoli will be larger in size. So smaller alveoli will have a tendency to drain into the larger alveoli and collapse. Now the role of surfactant is it will prevent this condition by ensuring equal stability for all the alveoli, and this will ensure that there is even distribution of ventilation to all parts of the lung. So that comes the second function of surfactant. Moving on to the third function, it helps to prevent pulmonary edema and ensures that the alveoli is kept dry. Now imagine a condition where surfactant is absent. So in this condition, the surface tension will remain unopposed. There is no force to oppose the surface tension. So what happens is there will be transudation of fluid from the blood vessels into the alveoli, and this can lead to a condition called pulmonary edema. Now with the presence of surfactant, this condition is prevented and surfactant will ensure that the alveoli is kept dry. Now next function is surfactant will also help to decrease the work of breathing so that the breathing mechanism becomes easier. And the last function is it provides innate immunity. Now we have already seen under composition that surfactant is composed of apoproteins and immunoglobulins. So these apoproteins and immunoglobulins, they can act as opsonins. So by acting as opsonins, they can cause phagocytosis of the bacteria and viruses and by the alveolar macrophages, and thereby provide innate immunity. So this slide summarizes the important functions of the surfactant. It helps to reduce the surface tension, maintain alveolar stability, prevent pulmonary edema, then decreases the work of breathing and provides innate immunity. Now we will see the application of Laplace law in lungs. Now what is Laplace law? We know that Laplace law states that P is equal to 2T by R, where P is the distending pressure, T is the surface tension and R is the radius of the organ involved. Now in case of the lungs, during expiration, the alveolar radius has a tendency to decrease. So according to Laplace law, when the denominator is decreasing, the numerator should also decrease so that the distending pressure P is not affected. So this surface tension, that is the tension in the numerator, is mainly reduced by the surfactant. So surfactant will alter the surface tension appropriately to prevent the collapse or over distension. Okay. So you can see a diagram that is depicting the difference without a surfactant and with the surfactant. With the surfactant, even when the radius of the alveoli is altered, the surfactant will make sure that necessary adjustments are made to reduce the surface tension so that the distending pressure remains unaffected, and this will ensure the prevention of collapse of the lung. So this is the application of Laplace law in lungs. Now moving on to the last part, that is the clinical importance. So we will see few conditions where the importance of surfactant comes. One clinical condition is infant respiratory distress syndrome. There are other terms for this condition. It is also known as hyaline membrane disease or neonatal respiratory distress syndrome. Now this is a condition that is seen in premature infants, that is infants who are born before attaining complete maturity. So in premature infants, what happens is the surfactant synthesis will not be complete. Usually complete surfactant synthesis happens around 31 to 32 weeks of intrauterine life. Now in premature infants, the surfactant synthesis will not be complete, so the lung maturity will not be complete. Okay. So surfactant is required for the proper expansion of the lungs. In premature babies, incomplete lung maturity can lead to the failure of lungs to expand. So this can lead to the instant death of the infant. Another reason could be the prolonged immaturity of the epithelial sodium channels. This is another factor contributing to the hyaline membrane disease. Now how can you diagnose this condition? One method is prenatal estimation of the lecithin sphingomyelin ratio in the amniotic fluid. Now suppose if the ratio is less than 1.5, it indicates high risk of developing hyaline membrane disease. Okay. Now treatment involves synthetic or bovine surfactant preparations are available in the form of inhalation. You can either administer them or you can give glucocorticoids or steroids to the mother. This will promote the surfactant synthesis. Okay. Now the adult counterpart of infant respiratory distress syndrome is ARDS, that is adult respiratory distress syndrome. Okay. Now the conditions which can predispose to this are chronic cigarette smoking, then long term inhalation of 100% oxygen which can lead to oxygen toxicity, and we have already seen that oxygen toxicity will inhibit surfactant synthesis, and occlusion of one main bronchus or one pulmonary artery. These are few conditions that can predispose to adult respiratory distress syndrome. So we have seen infant respiratory distress syndrome, which is also called as hyaline membrane disease, and you have adult respiratory distress syndrome. Now another condition that is having surfactant importance is atelectasis following surgery. So normally during deep breathing, the lungs can inflate to larger volumes and new surfactant molecules will spread thoroughly on the alveolar surface. But with shallow breathing, the spread of surfactant is often impaired. So when a person is recovering from surgery after anesthesia, what happens is we will often advise the subject to take deep breathing. So this will ensure even distribution of the surfactant molecules through the alveolar surface, and this helps in the speedy recovery of the patient after anesthesia. Okay. So with that we have come to an end of the topic. So to summarize, today we have seen what a pulmonary surfactant is. It is nothing but a surface tension reducing agent. Then we have seen the structure and the source of the surfactant. So the primary source of the surfactant is from type 2 pneumocytes, and the main component of surfactant is dipalmitoyl phosphatidyl choline. And from where are these surfactant molecules stored? They are stored within the lamellar bodies. Then we have seen the important functions of surfactant, like it helps to reduce the surface tension, maintains the alveolar stability, then prevents pulmonary edema, helps to reduce the work of breathing, and provides innate immunity. And finally, we have seen few applied aspects like what is infant respiratory distress syndrome, adult respiratory distress syndrome, and atelectasis following surgery. So with that, we have come to an end of the class. Thank you.